Welcome everybody to this week's seminar. This is supposed to be hosted by Dr. Indika Rajapaksa. I'm a student, Stephen Lindsley, in his lab, but something came up and he's running a bit late, so I'll have the pleasure of introducing our guest this week, Duncan Ralph. So Dr. Ralph attended the University of Santa Cruz for his undergraduate studies in physics, completing his thesis on energy transport and condensed matter theory in 2005. He completed his PhD at MIT in 2014, working on the Large Hadron Collider at CERN. And his thesis described the observation of the Higgs boson decays to 4 leptons. Since 2014, he's worked in Frederick Motson's lab at the Fred Hutchinson Cancer Research Center, first as a postdoctoral researcher, and more recently as a staff scientist, writing new computational methods for the analysis of B-cell receptor deep sequencing data. So it's my pleasure to turn over the presentation to Dr. Ralph.
Okay, thanks a lot, and thanks everyone for having me. Let me know if there's any problem with my audio or the background or anything like that. And I think questions is just interrupt and unmute if that works for everyone, and yell loudly if I don't hear you. Okay, start sharing my screen. Okay, you should be able to see everything there. Okay, so just need to start my timer. So I'll be talking about a variety of different inference methods that we've been writing over the last few years that you infer a lot of interesting things on B-cell receptor deep sequencing data sets that use pretty heavy use of hidden Markov models in a lot of cases. So first I'll describe a little bit of the structure of how the immune system works and how antibodies fit into that structure. And then I'll talk a very little bit about how we measure this with sequencing data. But mostly I'll be talking about 3 of the main inference methods that the software package that all of this is in, called partis, uses. So first, I'll talk about grouping sequences into clonally related families that stem from, each of which stems from a single naive rearrangement event. And then I'll talk about, so in newer data, we now have like pairing information about the heavy and light pairs that make up an antibody. And I'll talk about how we can use this information in newer data to improve the clonal family clustering from the first step there. And the final major piece I'll talk about is how we can predict antibody affinity just from the sequence of the BCR receptor. And then for all these, I'll be talking about how we've applied them to real data sets that we actually care about. So one way to think about the main goal of what your immune system is trying to do is that it's trying to kill things that aren't you. And there's 2 main parts to your immune system. There's the innate immune system and the adaptive part. The innate immune system is much older. We share it even with fungi and plants, kind of an innate immune system. It's generally fast and not very specific to particular pathogens. Typically might respond in minutes to hours, and one example of it responding is just you swelling up when you get a cut or a bruise or something like that. The adaptive immune system, on the other hand, is generally very specific to particular pathogens. And at least in the first exposure, is very slow. So antibodies, for instance, you start to build up significant quantities after maybe a couple of weeks. On subsequent exposures, the adaptive system responds quite a bit faster. And this is the foundation of immune memory, which is the reason in many or most cases, you don't get disease from a single pathogen, from the same pathogen twice. And also, of course, this is how vaccines work. So antibodies are part of one of the main parts of the adaptive immune system. I'll be talking about them in the rest of this talk. So 2 ways to I'm showing here 2 ways to visualize just the general structure of what antibodies look like. It's kind of shared with all antibodies. On the right, this is just a space filling diagram. And on the left is the same thing, but showing a few more labels here. So it's a dimer, and each half of that is composed of a heavy chain and a light chain. And both the heavy and light chain contribute to the main goal here, which is binding to a particular antigen. And the heavy and light have a lot of differences, but they're basically analogous in everything they're doing. So the way I like to think about the main goal of antibodies is that they're identifying or tagging things that we don't want to be there, that we want to get rid of. And once those things are identified or tagged, maybe the antibody actually does the destroying by directly neutralizing, for instance, or maybe the antibody signals to something else in the immune system to get rid of the thing. But in either case, we generally want antibodies to be extremely precise. So they bind to a specific piece of a specific pathogen, like a specific viral variant. And this precision is really important because, I mean, in a lot of cases, if you have antibodies that aren't very specific, that bind to several different things, that can be the source of a lot of diseases. So we want antibodies to be able to very specifically bind to a particular pathogen, but we also need antibodies that can cover the entire space of potential pathogenic threats that we might encounter. And this space is kind of, it's basically like the space of all possible peptides that are like 15 or 20 amino acids long. So those is, of course, incredibly large and incredibly high dimensional. But one way I like to visualize this problem is in 2 dimensions here, where I'm imagining that these 3 red squares are pathogens that we might be exposed to. And the green crosses scattered around here are BCRs or antibodies that we have available to respond to them. So you can imagine that your immune system would want to kind of evenly populate the space with the green crosses. And because this hypothetical space of potential antigens is so impossibly large and high dimensional, the amount of information necessary to code for that kind of diversity directly is just many, many, many orders of magnitude greater than you have in your genome. So this diversity generation happens somatically after germline. And so the first step of B cell development that generates this diversity is called BDJ rearrangement. Those 3 letters are just arbitrary labels for gene families. And so this bottom sequence here is the complete BCR sequence that codes for the antibody. And the way your body constructs that during B cell development is that you have a bunch of different V genes, like around 50 different kinds of different V genes. And one of these V genes will be chosen here. And you also choose 1 D gene and 1 J gene. And then you join the 3 V and D and J genes together. And as you join them, you delete some random number of bases from the boundaries. And you also insert some random number of non-templated nucleotides here in blue. So the end result of this is a naive BCR sequence, which means like one that, so this is a B cell that is not yet encountered its cognate antigen. And this picture, so this is for the heavy chain. The light chain is basically the same, but there's no D gene. So this diverse region is much smaller. And tying together the last 2 slides. So this is that diverse region, and that's called the CDR3. And as you might perhaps guess, like the CDR3 is the diverse part, and it ends up being the bit that's going up against the antigen, so it's determining complementarity. So BDJ rearrangement is kind of the initial diversification step that gives you kind of an initial rough draft. Affinity maturation is the next step that kind of refines that diversity. And affinity maturation is a selective process where there's a mutation going on. You start out with, if you're a naive B cell at the top here, so these colors are representing your BCR sequence. So like the green is the particular V gene you have, the orange is the D gene. And then we're imagining that this BCR codes for an antibody that's represented here, or a BCR that's represented here on the surface, where the shape of this is in CAR2 form, representing the specificity. So we're imagining that some antigen comes in and it binds well enough to this naive B cell that it's activated. So when a B cell encounters its cognate antigen, it's given permission to migrate to a germinal center and then start reproducing to create offspring. And when B cells reproduce in germinal centers, they purposefully introduce a huge amount of mutation right at the BCR locus. It's about 10 to the 5 times the background rate in your germline. Because there's a selective process where offspring that have higher affinity for the antigen have more offsprings. The way we represent this here is, so this naive B cell has 2 offspring. This one on the left has a red mutation in the blue bit here, and this other one has a red mutation in the purple bit. And we're imagining, so we're imagining that this mutation changes the antibody structure here to fit the antigen a little bit worse, whereas this one changes it to fit the antigen a little bit better. So since this one fits a little better, it goes on to have another offspring that has another mutation here that changes the antibody or the BCR to fit the antigen even better. And if we decartoonify this picture, there's of course many more B cells and a much longer process over weeks or months. But still, so at the end of this process, you end up with antibodies that are typically, or at least a lot of the time, several orders of magnitude higher affinity than the original antibodies. So tying together the last few, the pictures in the last few slides, on the left here, we have BDJ rearrangement at the top. It's kind of doing the rough draft of diversity. And affinity maturation in the bottom is kind of refining that. And the way I tie this into the two-dimensional representation is that VDG rearrangement is choosing these green crosses. So where these green crosses go and like trying to evenly populate the space. And then affinity maturation is represented as we're imagining that this red square, which is a pathogen, that we were actually exposed to that. And then when we're exposed to it, the nearby B cells, the nearby green crosses reproduce and go through affinity maturation, which is represented as this tree that's kind of like their offspring evolving to be closer to the red square. So evolving to have higher affinity for the red square. So before we talk about measuring these processes, I just have a couple of examples on the slide about things that we're generally interested in learning from these processes. So first, we can imagine that this might be an antibody that we know is really good against a particular pathogen. And we might want to know, like, is this an antibody that most people can make or not? So one of the main contributors to that is the V gene it uses. So the V gene locus is incredibly diverse between people. It's similar to the MHC locus, which you might have heard about more. So there's about 50 V genes in each person, and you probably share between 1/4 and 1/2 of those with a person sitting next to you if you're unrelated. So in terms of this, if we know that an antibody or an antibody class requires the use of a specific V gene, some V genes, everyone has. So that would be a great antibody to try and target with a vaccine. But there's lots of other antibodies that they use V genes that only a couple, like 10% of the population, or maybe even only a particular subpopulation in a particular part of the world have. Another thing that we might be interested in learning is, the way you respond to a particular flu strain is kind of unique in that it's very heavily dependent on your exposure history to different flu varieties in the past. This is called antigenic seniority. And this relates to the antibodies that are responding to that, in that if we're interested in finding out why a particular antibody in a person that was highly effective against flu developed, it'd be very important to know what the flu strains they were exposed to in the past. And the final example here is something that I'll actually be talking about a lot in this talk, where this is where we're imagining this. We know this antibody here is highly effective against neutralizing HIV. And most HIV neutralizing antibodies have really high mutation levels just because HIV is a really effective virus that usually takes a lot of mutation to find an antibody that's broadly neutralizing. So that means that this is a really long path. And one way that I'll talk about a little more later that we might be able to create an HIV vaccine is by recapitulating all these steps with a sequential vaccination. And that's the kind of thing, in order to do that, we need to have a really good understanding of this mutational pathway here. Okay, so when we want to observe the processes I've just been describing, the main tool that at least is relevant for this talk is deep sequencing. And we've been doing, not us personally, but just as a field, we've been doing lots of deep sequencing on BCR repertoires for the last 10 years maybe. And this gives you a really large subset, maybe 100,000 of the sequences in the body. And it is large, but it's important to keep in mind that there's only maybe 1 out of 10 to the 5th of the B cells alive in your body at any one point in time, and probably only maybe like 1 in 10 million or 1 in 100 million of the B cells that are alive in your body over the course of months or years. And so the way I'm kind of representing this data in this cartoon here is that, so in the data, the sequence here, the BCR sequence is just gray now. So that we don't know, showing that we don't know what V and D and J genes were used. We don't know the deletion boundaries or the insertions. And also all of the sequences are just smashed on top of each other into a morass because we don't know which ones are clonally related. And we also don't know the phylogenetic structure here. So what we want to do, and what I'll be talking about in the rest of this talk, is going back here to infer some representation of the reality that's occurring. So the first step in this is to group together the sequences that are clonally related. I'll call clonal families. And what I mean by that is sequences that stem from a single naive rearrangement event. So by definition, clonally related sequences have identical V and D and J genes and exactly the same deletion boundaries and also inserted bases. So given that, you might imagine the naive way to cluster these together, and indeed what's basically the standard approach that the vast majority of practical papers still use, is to group together sequences that have identical V and J genes. You don't use the D because it's just much harder to infer that accurately because it's so short and there's so many deletions. So you require the same V and J gene, and then you do single linkage clustering with a nucleotide similarity threshold within the CDR3, which is the most diverse region of the antibody. The problem with this approach is if there's any kind of significant SHM or any significant mutation, the accuracy is just absolutely terrible. The first problem is that if your gene inference is wrong, which it is a lot of the time, or at least there's a lot of uncertainty in it, then the clusters will be wrong. The other problem is that it conflates, on the one hand, somatic hypermutation, and on the other hand, naive sequence inference inaccuracy. And this figure here, I think it may have had a little bit too much adding a lot of detail to it. But I'll just focus on one aspect. This is kind of explaining the last thing I said, and that, so we have this true family here with a bunch of black dots, which are the observed sequences, and it's showing that if we cluster with these red dashed circles on here, just based on the observed mature black dots, we're going to spuriously, erroneously split apart this family. Whereas if we were just doing this like simple-minded single linkage clustering, if we use the inferred ancestral sequences here, even though these are not perfectly inferred, we would do much better. So what I mean by conflating is that we're conflating this distance with this distance. When if we're doing clustering, we really don't need to spread them apart by this distance. So an approach that we think makes a lot more sense for this is to use hidden Markov models. One reason is that these let you incorporate all the details of VDJ rearrangement and somatic hypermutation. And also, equally importantly, it lets you integrate out your annotation uncertainty, so your uncertainty about what particular V and D and J genes and deletions were used. So just in general, there's a lot of uncertainty in a lot of cases about what the annotation was. And if you care about clustering, you don't want to tie that clustering to a particular annotation. One way to visualize what an HMM is doing is by analogy to a normal distribution. So on the left, you can think of a normal distribution in this box as something where you put in an X value and turn the crank, and it spits out this Y value, which is more or less the probability of X, given that the normal distribution is the right model, and given that these two parameters, the mean and width, have the correct values. So looking at the right, an HMM, it's way more complicated, and it has many, many more parameters, but it's still doing basically the same thing, in that you put in a BCR sequence, and what we get out of it is the probability of a particular annotation or a particular clustering or something like that. Zooming way in on the HMM to see what it looks like. So what an HMM consists of is a series of states. So in here, each of these open circles is a state. So an HMM consists of a bunch of states together with rules for transitions between them, where each transition has a probability associated with it. And also each state has rules for which bases it can emit together with probabilities. So a probability of emitting A versus emitting C, et cetera. And so what you do when you pass a sequence into an HMM, the sequence is a series of A's and C's and G's and T's. And as you move along that sequence, you are moving along the sequence of states, deciding which state to go to and deciding what base to admit at each point. So that gives you a path through the HMM, which is this red line. And if we zoom out, so this is the whole HMM that's representing VDJ rearrangement. So a path through this HMM is equivalent to saying this sequence, it's a particular naive rearrangement or a particular annotation. This particular path, it's chosen this V gene, and it goes along the V gene and jumps out a little bit early, so there's a deletion here. It jumps into this non-templated insertion state and loops around a few times to insert a few bases. And then it jumps into this D gene and skips a little bit at the start, which is also a deletion, and then goes on to do entirely analogous things over here. So this, just to say it again, this path represents a particular annotation or naive rearrangement, and the HMM has a rule for just giving you the probability associated with that. So if all you're interested in finding is the most likely annotation for a sequence, you just choose the most likely path. And there's super well-established dynamic programming algorithms. There's the Viterbi algorithm that can look through all the paths and find you the most likely one in a very efficient manner. For clustering, what we're more interested in is the total probability of the sequence given the model. And this is just, you sum the probabilities over all possible paths. And happily enough, there's a nice dynamic programming algorithm called the forward algorithm that also calculates this very efficiently. The last piece we need before we can look at clonal relationships to this is to represent how we can have two or more sequences be clonally related in this HMM. And the way we do that is just force the sequences to take exactly the same path through the HMM. And we call this the multi-HMM because at each point, the only difference is that then we're omitting 2 bases or 3 bases because we have 2 or 3 sequences there. So to decide whether 2 sequences or 2 clusters are clonally related, we just form this, this ratio here, where in the top is the total probability where we've forced all the sequences to take the same path through the HMM. And in the denominator is the total probability, well, the sum of the total probabilities where we force them to take individual paths through it. And to use this for clustering, you do hierarchical conglomeration, which is shown on the bottom here, where you first merge together the 2 nearest sequences, and then the next 2 nearest, and then the 2 nearest clusters, and so forth. And you keep going until this ratio for all pairs falls below 1. So when you want to go and measure how well this kind of clustering method is doing, typically you have to do most of your validation on simulation, because on real data, one problem is that you almost never know the right answer. And the other problem is you just never have very much real data. So to really have confidence in a method like this, you need to test the method in all regions of parameter space. And the data really only exists at like a few isolated points. So you need to make simulation to really do a good job of spanning the whole space to do this. So this is what we're doing here. This is showing clustering performance on a variety of simulated repertoires, where the variable we're scanning across here is kind of the clonality of the repertoire. So on the left side of the x-axis is a very diverse singleton repertoire. On the right side is a much more highly clonal one that consists of just a few large families. And on the y-axis is the F1 score, which is what we're using to measure how good the clustering is. And it's just a number between 0 and 1, where 1 is perfect, that encompasses both overclustering and underclustering, so sensitivity and specificity in one number. And one other thing before is that, on the left is showing repertoires that have typical mutation levels, and on the right is ones that have much higher than normal mutation levels. And that's important because kind of in any BCR inference problem, the mutation level is just this knob where you turn it down to 0 and the problem's totally trivial. There's really nothing to do. And you crank it up high enough and the problem's just impossible. So this is kind of just 2 regimes where one is like typical / easy and the other one is like much more challenging. So the red line here is the HMM-based method, partis, that I've been talking about. And in the easy, for the easier inference environment, it's bumped up near the top, which is nice. And then when you move to the more challenging environment, as you expect, it moves downward and has worse performance, but it's in a fairly well-controlled way. It's still, it stays fairly horizontal there. And the main method I want to compare it against is the light blue called the VJ-CDR3, which is the kind of naive approach that's by far the most common in practical papers still, is light blue. And as I was kind of alluding to before, the problem with this is that it dramatically oversplits anything with significant SHM. And one way to see that is that for repertoires on the left side here, where the true answer is just the singleton repertoire, the light blue does really well, because if you oversplit everything, if the correct answer is to split everything into singletons, then you'll do really well. The other thing just to mention is that you can see what I was saying earlier, I think, that when you have higher mutation levels, it does really particularly poorly. So it gets close to down to the bottom here, which is just random chance, because it is so gratuitously oversplitting things. So once you have clonal family groupings, the other main step we need to get back to some representation of reality here is to do phylogenetics. And the way I drew this original one, this is making it look like we're sampling all of these ancestors here. And probably a more typical case is where we're mostly sampling leaf sequences, and then we need to use the phylogenetic program to infer these sequences here that are grayed out. And there's many, many, many different phylogenetic programs out there. The ones that are most relevant for this are ones that take into account the 2 main peculiarities of BCR development. The first is that the mutation is context dependent, which means that the mutation rate doesn't just depend on the base at a particular position, it also depends on the bases on either side, the 2 or 4 bases on either side. And also, you usually want the program to also take into account that the mutation process might be different in the V region to what it is in the J region. And the program that we really like to use for this is called Linear Ham. This was written by one of our students a couple of years ago named Amrit. It's a Bayesian phylogenetic hidden Markov model. So it's using an HMM it's a totally separate HMM to what Partis was using. And you can hopefully tell from this picture, it's simultaneously inferring the annotation here at the same time as doing trees. And the fact that it's like fully probabilistic and Bayesian, it means that you can sample from posterior, what it's doing is sampling from posterior on trees and annotations and inferred ancestors. And this is really useful for what I'm going to show you later. So a lot of the data we've been running on and using in actual papers with biology net has been made by Julie Overbau's group that's also at the Fred Hutch here. And most or all of this data has been from a particularly unique cohort of HIV samples that Julie was involved in sampling from Kenya 20 or 30 years ago. And the reason these are very interesting from the perspective of trying to develop an eventual HIV vaccine is partly because they're from before the widespread availability of antiretroviral therapy. So the infections are just much more uncontrolled than they would be today. And also it's a very high risk cohort. So there's a fair amount of super infection with different clades of HIV. And both of those things are associated with being more likely to develop really good broadly neutralizing antibodies that can neutralize a bunch of different clades of HIV. So the commonality between all the papers I'll be showing here is that Julie's group, in this case, Laura and Cassie, they go and take they unfreeze some samples and they go and use wet lab techniques to pull out a bunch of really good antibodies by some criterion. So here, what they're doing is using microneutralization. This is just the X and Y axes are two different replicates of this. And the ones they're choosing to go test more thoroughly, I think is all the dots in this plot, but the ones that are highlighted in blue are the ones that turned out to be really good. And then the next step is to unfreeze some more blood, and they do deep sequencing on that. And then they pass that to us. And what we do is for each of these antibodies that turned out to be really good, we pull out all the clonally related sequences from the deep sequencing data and then analyze those families. So this is just a simple scatter plot showing all those families in mean SHM versus the size of them. And there isn't a lot to take away from this, but you can just in the simple plot see that the families of interest, which is where the good antibodies are, do tend to be in larger families and have higher levels of mutation. And just quickly, the reason this particular data was very interesting to us is because this is from a mother-daughter pair where vertical transmission occurred, which means that the mother infected the infant after birth. And this is, besides being super tragic, this is very interesting from the perspective of potentially developing an HIV vaccine, because in most HIV transmission events, the person who is getting it is totally naive to HIV. They don't have any HIV antibodies. But in vertical transmission, because just generally speaking, mothers transmit antibodies, like share their antibodies with their infant, especially through breast milk, that when there is a vertical transmission event, the person who is acquiring HIV already has a bunch of antibodies to it that are potentially very good, which is much more similar to the case that you would have if you had been vaccinated for HIV and then were exposed to it. So talking a little bit more in detail about this hypothetical route to an HIV vaccine, the way this would work in theory is that you would find an antibody that you know is really good in broadly neutralizing. So BNAB is the abbreviation for that. So you'd find a good antibody using wet lab means, and then you'd do some deep sequencing and reconstruct the clonal family, and then use something like linear ham to reconstruct this particular evolutionary pathway between these inferred ancestors back to the unmutated ancestor, the naive sequence way up here. And in the next step, you would design some antigen that is designed to elicit the particular unmutated ancestor here. And then you design a series of booster immunizations along here, where each one is designed to to nudge these ancestral antibody sequences in exactly the proper direction to recapitulate this mutation path so that you end up eventually back at the mature BNAb that you were aiming for. And at least to me, this sounds very cumbersome and kind of unrealistic, but that people can actually do this with mice. So it is something that is actually being done, at least in the terms of a proof of concept level kind of thing. So this is a paper where I'm showing a little bit more of the development, going a little further with the development. So again, here, Cassie and Laura have found a bunch of antibodies that they know are highly neutralizing, highlighting one here in blue called BF520.1. And then they do deep sequencing, and we pull out the related sequences and make trees with them. And then we run linear ham to give us a probabilistic view of what all of the inferred intermediate sequences and the important mutations were that happen in that development path. And that's summarized in this flow chart here, where at the top we have the naive sequence, and at the bottom the mature sequence, and each oval here is an inferred ancestral sequence. And the brightness of the red is how confident we are that it was really there. And the darkness of the blue arrows is showing how confident we are that that was a mutation that actually occurred. And the next step in this process is to pass the sequences back to them so that they can, of course, very labor intensively, go synthesize them and test the neutralization of these antibodies. And this kind of chart is really important to that because it gives you a lot of confidence which ones you really need to synthesize. For instance, this one is bright red, so we're really sure we need to do that one. But then when you get down to here, you have these two alternative pathways, and you need to test both of those to see which one is the correct one. The end result of that, we summarize in this table here, where on the left, you have the unmutated naive ancestor that it binds to HIV, I think I'm remembering that correctly, but it doesn't neutralize anything. And on the right, you have the mature antibody originally pulled out that has very good neutralization breadth and about 6% nucleotide SHM. And then as we go along the chart to the right, these are all the inferred ancestral sequences. And the really exciting thing about this is you get to almost full neutralization breadth with only a third or a quarter of the total number of mutations here. So this means that like the number of key mutations that were really important to acquire that neutralization breadth is actually really small. And because of that, and because if you have some friends that can do this magical structural stuff, you can look at exactly what the mutations look like on the structures. So here, this is a cryo-EM structure. It's showing the antibody binding to the relevant bit of HIV. And then on the right, if we zoom in to that contact surface, what they're doing here is they can really just visualize where the mutations in red. So exactly these key mutations, how they change the way that the antibody is binding to ONF and enable the broad neutralization. So most of the time, when we're talking about antibodies, we're talking about them acting via neutralization. And that a typical way would be that the antibody directly binds to the pathogen and stops it from entering the cell. An example of this being important is that you might have seen in the last few weeks, the meta-analyses coming out. So we have a really nice variety of SARS-CoV-2 vaccines now. And you might have seen some plots where like on one axis, they have the induced neutralization titer. On the other axis, you have the vaccine efficacy, and they can draw really nice, I mean, large confidence intervals, but a really nice best fit line through there. So in these cases, the neutralization is really all you need to predict how effective the vaccine is going to be. But this isn't always the case. Another way that antibodies can contribute to viral control is with something called ADCC, which stands for antibody-dependent cellular cytotoxicity. And this is illustrated here. We have a cloud of antibodies binding to something they don't like here. And then some other effector cell from the immune system is binding to kind of the backside of the antibodies, and then shooting laser beams at the cell and lysing it to destroy it. And a good example of a case where ADCC was really important is, and I think it was the largest HIV vaccine trial to date was the RV144 trial. And this was, I mean, the big picture result of this, it was not an effective vaccine, but it did achieve some level of efficacy, some level of protection. And when they look at the correlates of what kind of the correlates of protection that could predict whether it was protective, ADCC was a correlate of like if you had a strong ADCC response, you were more likely to have protection. But I think there was actually no correlation for neutralization. So in this case, at least ADCC seemed to be a lot more important than neutralization. So in order to relate ADCC activity to the inference to the inferred ancestors I'm talking about, I need to go into a little more detail about antibody structure. So this is the bit that I was saying is this is the variable part of the antibody that determines the pathogen specificity. And I mentioned the variable, there's a CDR3 region here. There's actually 3 CDRs here that determine the complementarity. And the CDRs, these are very variable with the blue spikes here. And they're interspersed with a few framework regions that are much less variable. And in this image at the top right, you can see why that is, because the CDRs, they're these loops that are hanging off and directly contacting the antigen, whereas the framework are much more buried in here in like the interior of the antibody. So keeping that in mind, in this paper, the first part is just the same as I've been describing in the other 2 papers, where in this case, Laura, they go and find a bunch of really good antibodies in these samples. In this case, they're looking for antibodies that have really good ADCC activity, not neutralization. Then they do deep sequencing. We pull out the families and infer the ancestors using linear hand, doing this kind of thing. Then we pass the sequences back to them. They synthesize them and test, in this case, the ADCC activity, not the neutralization, to determine which of these mutations were important for allowing ADCC activity. And we finally summarize what's going on, or what we think is going on, in this table here, where these along the left side are all of the antibodies they're testing. And then the columns, it's looking at like, where were the mutations? So in the left column is the mutations that were allowing just binding to HIV. And in the right column is the mutations that were allowing really strong ADCC activity. And we're looking at where in the antibody those mutations occurred. So if we just kind of like squint, I think it's fair to say that like the first column that's allowing just simple binding to HIV is mostly yellow. So those are mostly in the CDR, which is the bit up here that's determining the specificity. So that makes sense, is binding directly to HIV. On the other hand, in the far right column, that's the mutations that are giving you strong ADCC activity. These are more green and blue, which involves a lot more mutations in the framework region, which are more in the interior. And that makes sense, at least hypothetically, because the interior of the antibody, you could imagine to be more involved with how it would bind to this effector cell on the back side of it. Okay, so now we're going to put our method development hats back on for a bit. So antibodies have 2 parts here. They have the heavy chain and the light chain. And the locus coding for the heavy chain here and the 2 different light chain loci, the kappa and the lambda, they're on 3 completely different chromosomes. So that means that basically all the deep sequencing data we've gotten over the last 10 years or whatever, we've had no idea which heavy chain pairs with which light chain. And this is obviously a huge problem if you want to synthesize those antibodies, because if you have a heavy chain that you think is good and you want to go test it, you have to just choose a bunch of light chains more or less at random and hope they work. And most of them probably aren't even going to make a functional antibody, let alone have the affinity that you're looking for. Another problem with not having this pairing information is that it makes for really inaccurate clustering. The main reason for this is that the light chain just has so little diversity compared to the heavy chain. So in this picture at the bottom, I'm showing that heavy chain here, on average, has about 20 bases that aren't V and aren't J. So this is where most of the diversity is coming from. Whereas the light chain here on average has only 1 base. So like light chain diversity is really mostly just a matter of choosing a V gene and choosing a J gene. And the effect of this is that on any reasonably sized repertoire, you'll have lots and lots of cases where 2 different light chain rearrangements result in naive sequences that are experimentally indistinguishable. So you're just going to super overcluster the light chain in essentially all cases. And we can see this happening on our real data. Here's an example where I was showing the distribution of cluster sizes for the heavy chain in green and the light chain in red and blue. And we know these are the same clusters, so they should be basically the same. Really, it would be that the sum of the red and blue would be the same as the green, but they should be basically on top of each other if the clustering is going right. But instead, the light chain is trying to pretend to tell us that the light chain cluster is an order of magnitude bigger than the largest heavy chain clusters, which is clearly just ridiculous. Luckily, people have recently figured out how to do deep sequencing in a way that preserves the pairing information. The most common way that people are doing that now, I think, is with the single cell droplet technologies from 10x Genomics. But in any case, we've worked out a way that we can use this pairing information to basically correct the clusters of the heavy and light chains. In a very cartoonified form here, the way this works is that you start out with the single chain heavy partition and the light partitions. So these partitions have the same sequences here, except for they disagree, for instance, here on whether they disagree on whether C and A and B are clonally related. And there's a fundamental asymmetry in this situation in that if the light partition says they're all clonally related, that isn't really active information telling us that they are related. It's just saying it doesn't have information that they're not. On the other hand, here, when they're split apart, that means that the heavy partition is saying that, like, C sequence is really different than A and B, so they really probably are. So this means that, like, when we want to correct, like, kind of join together information from these two, what you want to do is basically apply the splits from one partition to refine the other partition and vice versa, so that the joint clusters you get out of this are going to be the splittiest ones, so the splittier ones from both cases. And then to use this in practice, there's some fairly complicated steps to incorporate these joint clusters back into the partition you're making that has a bunch of overlaps that I'm just going to completely gloss over here. But the bottom line is that when we test this on simulation samples, we can see that it's doing what we want it to, which is basically correcting the heavy chain partition with the light chain information and vice versa. And I'm skipping over all of that simulation to just show you some results on real data where we don't know the correct answer, but we can still see that the method is basically behaving how we expect it to. So the first thing, so this is heavy on the left and the two light chains on the right. And the first thing is that the single chain partitions in green, initially they start out in the light chain being super, super over clustered, so going out to the right. But then as we apply the paired clustering method to get the red line, it's much more, the heavy chain and light chain partitions are much more compatible with each other. And the other thing to notice here, so it makes a massive difference on the light chain, but even on the heavy chain where, on smaller clusters it's not making a huge difference, it does still make a really significant difference to the largest clusters here. And at least for the kind of biological applications that I'm showing, the largest clusters are the ones we really care about. So having something like this, where a third of the sequences in your large cluster are spurious, that means you've probably got a whole lineage in the tree that isn't actually supposed to be there, which means it's really going to screw up your ancestral inference. Okay, and the last methods thing I'll talk about here is a way to choose antibodies that are likely to be high affinity based just on, like, the sequencing data. So in all of the HIV papers I was mentioning, they use wet lab techniques to extract PCR sequences or antibodies that they know are highly neutralizing or have a lot of ADCC activity. And there's a lot of different ways to do that. You can use cell sorting or microneutralization or that kind of thing. And all of those are really effective, but they're also really expensive and really time consuming. And it would be really great if there was a way that we could just use the sequencing data to predict which of those sequences in the deep sequencing data code for high affinity antibodies, because a lot of times we already have that data sitting around and we know some of those antibodies surely code for some of the sequences surely code for high affinity antibodies. The basic handle that we would want to use here for predicting which of these antibodies are high affinity is the evolutionary selection information. So affinity maturation is this process where, like, it evolutionarily just, like, applies selection to increase the affinity of the antibodies, and that should leave traces that we can extract from the inferred lineages in the final product. So in order to try and see if this kind of thing could actually work in practice, well, the first thing we put together, which again I'm just kind of basically skip over here, is put together this big simulation framework that can simulate all the relevant processes inside the germinal center and, like, the selective effects of affinity maturation. And then once we have that, we can test a bunch of different metrics here that I won't go into in detail, but there's a bunch of different metrics that we kind of test to see can this metric predict the affinity based on the evolutionary information in the tree. One metric I will touch on quickly because I can draw a nice picture for it, is called the local branching index. And this, it measures kind of the branchiness in the local area of the tree around the node or around the antibody you're talking about. And the idea is that if you're a high affinity B cell, you're likely to have more offspring. And that means that the area of the tree that you're in will have a bunch of branching going on. And this metric has been used a lot in the context of viral evolution to predict which flu strains are going to predominate in future years. Now, also, just if you see, I've got a big list of variables here, and I don't know which are good, and they each have some independent information, but it's not clear how much. The natural thing to do is to throw them into some kind of machine learning approach and try and get that to work well. So we sunk a ton of time into doing this. Using a decision tree regression turned out to be the approach that was seemed to be working the best. And this is just a chart that I won't mention, that I won't like go through in detail, but it has a bunch of the input variables. And at least to me, the most interesting part of this whole exercise was that it didn't work that well. We had no trouble training a DTR that can do about as well as each of the input variables, but we completely failed to make one that did significantly better than any individual variable. And we're pretty confident that this is just because of like the properties of how tree space works. So tree space, like the space of all possible trees, is like another one of these like spaces I like talking about that like you can't really imagine using it, but it's very complicated and high dimensional. And the problem is that like, you can train a machine learning approach easily so that if it knows exactly where it is in tree space, it'll have extremely good predictive ability. It'll work really well. But the thing is, if I just hand you a single tree, there's no way you can infer accurately enough the parameters of that tree to tell the DTR exactly where it is in tree space. So one example of that, so one parameter that's really important to this is the selection strength. And there's a lot of different ways to parameterize it. But this is basically quantifying the relationship between, if you have really high affinity, how strongly is that linked to the number of children you're likely to have. And this is really important for the performance of these metrics because there's some metrics that if the selection strength is high, they work really well. But if the selection strength is 0, if you're just mostly in neutral evolution, that metric can be totally useless. So in order for the DTR to function well, it really has to know what selection strength is. And there's lots of really fancy phylogenetic ways to calculate the selection strength, like the FAWH here is one of them. But none of them are anywhere near accurate enough to be able to calculate on a single tree where you are in the parameter space, which means that the DTR has no idea where it is. So anyway, if you go through all of the simulation validation on this and kind of check what different metrics work, happily enough, it turns out that almost basically the simplest method you could think of works better than all the others. So this is the consensus sequence. So for each family, it turns out that the sequences that are closest to that family's consensus sequence are most likely the highest affinity. And in retrospect, this is not particularly surprising, I think. Like the picture I have in my head is that the consensus sequence is each family's best guess for like the best antibody so far. This is the best antibody we've found so far. And then the family exists as this cloud around it, exploring the nearby antibody space, trying to see if there's a better sequence nearby it. And then another metric I'll mention just quickly because it's terrible is the number of mutations. So I mentioned this just because in a lot of practical papers, it's really common to take a bunch of antibodies, and when you want to choose some high affinity ones, just choose the ones that have the largest number of mutations. And on the face of it, of course, this makes a lot of sense because I just told you that affinity maturation is this mutation process that on average, applies a bunch of mutations and to improve the affinity of the antibody. The problem is that that statement is different to saying that within any group of antibodies, the ones with the most mutations will have the highest affinity, because the ones that have the most mutations, those are by definition leaf sequences. So by definition, they have a bunch of novel mutations that have not yet been subject to significant selective pressure. And in any context, most novel mutations are deleterious. So if you want to find the highest affinity or the fittest, highest affinity antibodies or the fittest bit of the tree, it's true you don't want to be at the root of the tree, but you also need to go back some ways from the leaves. And not coincidentally, that's basically what the consensus sequence is doing. It's some perhaps overly heuristic, non-optimal method of finding some middle ground, some compromise between the root and the leaves. So when we want to, if we want to test this antibody choosing methods on real data, we run into the usual problems with validation on real data, and that the sample sizes are really slow. You don't have a lot of data sets. And here the sample sizes are slow for the same reason that we're really excited about having this method, that it's just really expensive and time consuming to test affinity. So there's a million public data sets out there that test affinity or neutralization on a handful of antibodies. These are just the two that I have in the paper. These are two well-known HIV BNAB data sets. One of these I think has like 7 antibodies and the other one has like 15 or something like that. And the way I'm quantifying performance in these plots is that we're kind of saying, like, if I choose the best like 15% of antibodies based on my metric on the x-axis here, like what's the percentile in terms of affinities of those antibodies? So like if we choose the best in terms of a metric, are those also the best in terms of affinity, which is what you actually want? So the green lines here is showing that, you know, if we were perfect, this is saying that if we choose the top 1% using a perfect method, then we would of course also get the top 1% in terms of affinity. And the red dashed line here is random chance, because you just get, if you're choosing with blindfolded darts, then you just get 50% all the time. And just kind of looking at both of these are very small sample sizes, but you can see that like our blue line here is, you know, it's doing better than random chance here. And here it gets lucky and is actually just doing optimally. And it's doing fine. And as importantly, this is entirely compatible with the expected performance that we see in simulation. So this is a more recent thing. I don't have any like final results to show you, but we've been applying the paired clustering algorithms and the antibody choosing things with some really exciting data from Wesley Gu's lab. He's working on dengue, also at the Hutch. So we don't have those yet, but those will hopefully be kind of things that we're showing soon. So in summary, these B-cell receptor deep sequencing data sets, especially when combined with some wet lab techniques to like measure and find some good antibodies, they can tell you a lot of really important things that are really of biological interest. But you do have to be careful with certain steps in the inference process to really get actionable methods out of them. So like, first, of course, I'd like to thank Eric. He's been an amazing mentor for the last few years. And Amrit, I mentioned, he was one of our students, and he wrote the linear ham package that is really nice probabilistic Bayesian inference. Eli was a programmer in our group, and he wrote this amazing visualization package called Olmsted. There's some links in the backup slides. I'd be happy to talk about it or click through an example install. It's this really amazing visualization thing that we use a lot when we're passing data back and forth with wet lab groups, because it lets you click around on a web server and zoom in and out on trees and label the trees by different things while you're looking at the BCR annotations. And all the HIV data I was showing you is from Julie's lab. So working with data that Cassie and Laura and Mackenzie have made for the last few years, really been a great pleasure. And as I was just mentioning, the paired clustering methods and the antibody choosing is something that we've been working on with Wesley Gu's lab, also at the Hutch. So it's been really great working with data that Lisa and Jay have made. Thank you.
Stephen?
Yeah, thank you very much. It was a great presentation. Does anybody have any questions?
Yeah, hello, Duncan. This is Indika. Thank you so much, Duncan.
Yeah, thanks for listening.
Yeah, so Duncan, during affinity maturation process, is there like a common backbone to regardless of the antigen? Certain things has to satisfy to become a plasma B cell.
Is there a common what, sorry?
Like a common, I will use the word, so I'm talking mainly in the context of transcription factors.
Yeah.
So are there any common backbone regardless of the antigen that to satisfy to become a plasma B cell? So is that the somatic hypermutations are some sort of a refinement to
yeah, so I'm not let me know if I'm not answering the correct question, but I think what I'm understanding is went right past it. Yeah, so where's my framework picture there? So like, yeah, when you're having mutations in the B cell, well, yeah, lots of naive rearrangements, so lots of VDJ rearrangements.
Yeah,
I think maybe like 80% of them result in just failures. So like B cells that don't express, like maybe they have a stop codon, they're out of frame, or just they don't fold correctly.
yes.
And also when you have mutations, if the mutations, especially mutations in the framework region, those also result in like ineffective antibodies. Is that your question? No, sorry.
No. There's but I am thinking in the context of a naive B cell become a plasma B cell.
Yeah.
And during that process, it is like a differentiation, but normal differentiation, there is no somatic hypermutations, right? So in this B cell affinity maturation, you need to change the sequence, the small part.
Yeah.
So what my question is, the normal differentiation that certain things has to satisfy. Is there people know anything about that? Is there a common, so cell has to divide, right? And certain, like a common backbone to affinity maturation.
I think the fact that I'm confused means that the short answer is definitely that I don't know.
Okay.
The more speculative answer is that I think we don't know a ton about exactly how affinity maturation in the GC and class switch recombination work. And going to like the GC, like people, I mean, there's a few things that people agree on between plasma cells and memory cells. Like plasma cells tend to be higher affinity, memory cells tend to be more diverse. But beyond that, I think people are all just kind of like arguing about different stuff.
Okay. So, Duncan, by the way, I was a postdoc with Mark Grudin.
Oh, cool.
Long time ago.
Pipe in with a question here. Really nice talk. I'm curious, you mentioned, sort of one of the things that pollutes, your data or makes the phylogeny reconstruction difficult is that, like lots of these, mutated sequences might be non-functional. I suppose some are obviously non-functional by virtue of, having stop codon, nonsense mutation or something like that. But I'm wondering if, deep mutational scans or tight-seq or any of these kind of very high throughput experimental approaches, which can measure affinity, can those either kind of guide the inference process, or can you look at, over the course of affinity maturation, can you see like experimentally measured affinity going up kind of along the way at internal nodes?
Yeah, you definitely can. I guess the first caveat is that I'm very fuzzy on the big picture of exactly what experimental methods would be good for various things. But I can say that, like, yes, that is the kind of thing people are working on. The two projects that I think of immediately are, Gabriel Victoria's group. They do a lot of really amazing stuff with GCs. So so Will and Eric especially have been working with them. So that makes me think a lot of their work, and that those are like, small families, small sample size, because of course they're having to do all the work by hand. But that is where they're looking at each individual sequence and seeing how particular mutations change affinity. And the other project that sounds like, again, I think I'm not 100% understanding the question, but we've been doing a lot of, are you familiar with like FIPSeq and ViraScan? Yeah, so we've been doing, not me personally, but other people in our lab with Eric. So Jared and Kevin have been working a lot on FIPSeq data from especially SARS-CoV-2 with Julie's lab, with some people from Julie's lab, and I think Megan especially had a paper that was actually out. I could definitely point you to some papers on that, where they are, that is much more high throughput functional validation.
Okay, yeah, great. I mean, that's great. I'll check some of those out for sure. Thanks again.
I have a speculative question. In one of your slides, you had talked about gently nudging basically BCRs towards specific antibody targets.
Yeah.
In a series of steps, and I'm wondering, just generally, if you believe it's necessary to gently nudge, or if you might be able to push the BCRs towards that final state more efficiently or more aggressively. What are your thoughts around that?
I mean, I am very far from an expert on this kind of sequential immunization scheme for HIV, but I mean, I was just Googling trying to figure out which paper I was thinking of because I thought it was a recent paper that was like, oh, I thought I read this year that, oh, wow, someone actually managed to do this in mice. And then I started Googling it yesterday and I realized that, it's, they did it like 7 years ago for the first time. So, but that said, my understanding is that, I lost it. My understanding is that I think it's basically just a combinatorics thing. Like you can't, there's a lot of things in a person's, in your genetics that would determine like what V genes you have and where the mutations are likely to be. But to some extent, it's just kind of like you can't control. It's just this random process that decides exactly where the mutations go and exactly what V genes you start with. And if you have a process, if getting from here to here requires 10 mutations, like just combinatorically, that's some number to the 10th power, which is just a really, really big number to have in your denominator. Whereas if you have break it into a bunch of steps, that's just a smaller number of the denominator, so it makes it much more likely.
Yeah, totally fair. Thanks.
I actually had a quick question about the antibody-dependent cytotoxicity and why, or the data you were presenting that they were more likely to have mutations in the framework regions. And I was curious if those mutations in the framework regions could be used or are used for clustering clonal lineages and for like lineage reconstruction. Because I know in the past, you on previous slides, you mentioned you're basically focusing on the CDR3 region for that process. And I was curious if the framework regions come into that at all.
Yeah, so I mean, in all the clustering and inference, we use all the mutations, and we don't even really distinguish between the CDR and framework in terms of there. I mean, all the models have like per position rates for the mutations. So they know that like they would implicitly know that the CDR is more mutable than the framework region. But there's actually a really good point that in terms of there probably is some signal there, like some extra signal we could scrape out to like get some better performance by like taking into account like this is a CDR and this is a framework. So we know that fundamentally these mutations are not just that, I guess, not just that the mutations are less likely in the framework, but that a mutation in the framework is probably functionally doing something different than one in the CDR.
Okay, thank you.
I also have a question about the hypermutation. So are we assuming that the hypermutation always in the same rate in the whole process, or that could be changed? Like,
yeah, no, it's variable rate. It definitely changes a lot. And I guess I might be understanding, let me know if I'm not understanding right, but like, so one way in which, so one picture of the way SHM is happening is that, so I said you're a naive B cell, you're exposed to your cognate antigen, you go to a GC, and then you go through a bunch of cycles between the light and dark zone. So that's maybe like weeks or months. Your mutation rate of your offspring is like, maybe it's not constant, but maybe it's kind of constant while you're in that GC reaction. But then the problem is that like that GC is going to go away after some amount of time, and then maybe you'll float around in the blood for a long time. Or your offspring will as a memory cell, and then maybe you're re-exposed to the same pathogen and you go to a GC again and start it all up again. So there's this potentially very stochastic process where your mutation rate is really high for a while and then it goes down for a while. And this is actually, there's been some work on this. So in Ebola, you can have latent infections where the mutation rate will totally stop for a long time. And in some Trevor Bedford's plots I've seen where they have like, they have to take this into account. We have really dramatically varying mutation rates. So you end up with spuriously really long branches on the tree.
Okay, so so is it possible that in the same time constant and when the B cell divide, they could like use different mutation rate, right? I mean, yeah.
I guess it's kind of like, I'm just a little, just thinking on the fly. Like I'm used to thinking in terms of like, so a mutation happening on the micro level is this very stochastic process that happens individually. Like, an individual B cell is like, it's in the light zone. And then it's given permission to go to the dark zone and it's kind of going back and forth there. And in one of those, maybe mutation happens out of a generation. And then so like thinking about like the parameter in a phylogenetic model where you have the mutation route, there's kind of averaging over a bunch of cycles, and that, well, I guess the short answer is I haven't thought about that a lot, like how to parameterize that, but other people have certainly have.
I see. But in your like Hidden Markov model, that you assume that the rate actually is like 10% or 25%. Like it's
no, it's variable, but it is. So in my HMM, in the HMM is geared towards clustering. It's even simpler for most purposes, just assuming it's a star tree, just because that's what we have to do to make it fast enough to be able to run on the whole repertoire. And then if you do more detailed phylogenetic things, you're using something like linear ham that is much more accurate, but you could never run on 100,000 sequences.
Okay, I see. Thanks.
Okay, if there are no more questions, I think that might be the end of the seminar. Thank you again, Dr. Ralph, for presenting today. It was a great talk.
Yeah, thanks for having me. That was great fun.
Great. Thanks, everyone.